While psychology maps the universal machinery of the human mind, capacities like memory, language, and emotion, it also faces a second, equally critical objective, explaining the specific variance that separates one person from the next. For decades, clinicians relied on intuition to categorize these differences. Early tools like the Rorschach inkblot asked patients to interpret abstract shapes, but the results were often inconsistent. Testing the same person twice could yield entirely different scores, failing the basic scientific requirements of reliability and validity. Modern personality science moved past these subjective interpretations by aggregating thousands of descriptive traits from the dictionary. By filtering out linguistic overlap, researchers identified the 5 primary dimensions that define our character. This control board represents the modern consensus. Almost any question you could ask about a person's character maps onto one of these 5 settings: openness to experience, conscientiousness, extroversion, agreeableness, and neuroticism. In psychology, these are known as the Big Five, or Ocean. These settings remain remarkably stable over several decades and accurately predict how we live. Higher scores on the conscientiousness style correlate with greater marital fidelity, while people with high openness to experience are statistically more likely to change their jobs. By quantifying these 5 fundamental settings, personality becomes a tangible, measurable dataset that characterizes our style of dealing with the world. Beyond personality, psychologists focus on a second major dimension of human variance that dictates life outcomes: intelligence. While individuals possess distinct skills in math, language, or spatial reasoning, the data reveals a common underlying factor powering all of them. Researchers call this the G factor, or general intelligence. This metric correlates directly with the physical speed of neural processing and the capacity of working memory. When the central G factor is high, it tends to pull the submeters for specific cognitive skills up along with it. Standardized IQ tests are designed to capture this metric. Despite the complex social history of intelligence testing, these scores remain statistically accurate predictors of school performance and long-term career success. This leaves us with a measurable framework for the mind. The challenge for researchers is to identify exactly what turns these dials and sets their initial positions at birth. To find the origin of our traits, behavioral geneticists divide all human variants into 3 strict categories. There is heredity, or our DNA, shared environment, which accounts for our family home, and non-shared environment, which includes random life events and distinct peer groups. In this context, heritability describes what proportion of the differences between people is driven by their genes. It is a measurement of variance across a population, rather than a percentage of a single person's character. We see this in the number of legs a person has. While having 2 legs is a genetic trait common to almost everyone, the differences in leg counts across a population are almost entirely environmental, caused by accidents rather than DNA. Researchers isolate nature from nurture by comparing the similarities of identical twins, who share 100% of their genes, to fraternal twins and adopted siblings. These comparisons allow us to see how much of our character is set by our biology versus our upbringing. The most striking evidence comes from identical twins separated at birth and raised in different families. Despite their different environments, these adults show remarkably similar settings on their personality and intelligence styles, sharing everything from their religious attitudes to their brain structure. The high correlation between identical twins raised in isolation suggests that our psychological architecture is significantly influenced by heredity long before our environment begins to shape us. The data reveals that the shared environment, the influence of traditional parenting, accounts for a statistically near 0 proportion of the variance in adult personality. Statistically, 2 adopted children raised in the same home are no more similar to each other than 2 total strangers. We often see child effects, where a child's inherent genetic traits trigger specific parenting responses. A highly inquisitive child may encourage their parents to read more often, creating a correlation between parenting and intelligence that was actually driven by the child's own DNA. However, high heritability within a group does not rule out environmental effects between groups. If we plant identical seeds in 2 different plots, one fertilized and one barren, the height differences within each plot are genetic, but the massive difference between the 2 plots is entirely due to the soil quality. This environmental impact is visible in the Flynn effect. Over the last 70 years, global IQ scores have risen significantly, a shift far too fast for human evolution. This rise reflects structural environmental changes in nutrition, education, and technology that benefit an entire population. While our individual dials are set largely by our genetic code and random life events, the overall baseline limits of our society are dictated by the structural environment we inhabit. When our internal architecture is pushed past its structural limits, these settings become extreme deviations from normal human variance. This is the domain of clinical psychology. Psychologists approach mental illness using a medical model, categorizing disorders as severe, dysfunctional deviations from normal baseline traits. Treatment begins where our internal architecture fails to meet the demands of everyday life. Schizophrenia involves a cognitive breakdown, where thoughts can no longer be sequenced or coordinated, leading to hallucinations and delusions. This disorder is linked to high genetic heritability and specific neurochemical malfunctions, such as an overabundance of dopamine activity. Anxiety disorders like phobias follow a different logic, known as preparedness theory. We are biologically predisposed to fear ancestral threats like snakes or spiders, rather than modern dangers like cars or guns. Evolution left us highly reactive to the threats of the past. The diathesis stress model explains how these conditions emerge. A clinical disorder requires two intersecting factors: a latent biological vulnerability, followed by a high-intensity stressor in the non-shared environment. One provides the potential, the other provides the catalyst. This interaction suggests that a disorder is neither purely genetic nor purely environmental, but the result of a specific negative life event meeting a pre-existing genetic architecture. Because environment plays a role in triggering these states, clinicians use targeted environmental inputs, ranging from talk therapy to medication, to stabilize the system. Cognitive behavioral therapy identifies distorted thought patterns that have become systemic. By training patients to recognize and interrupt these cycles, the therapy provides a manual method for the patient to reset their own cognitive dials. When behavioral interventions are insufficient, direct medical approaches are used to recalibrate neural chemistry or electrical activity. For the most severe cases of depression, electroconvulsive therapy remains a highly effective method for resetting the brain's baseline state. Beyond specific techniques, the successive therapy often relies on non-specific factors like empathy and hope. When a patient believes a recalibration is possible, that expectation can act as a self-fulfilling prophecy, facilitating the recovery process. The human mind is a complex, genetically pre-wired instrument, that it is not a closed system. By scientifically measuring its variance, we obtain the blueprints needed to understand our differences and the tools required to repair the architecture when it breaks.